Yes, dear children, in the previous lecture I was telling you about the different amino acids, essential amino acids and non-essential amino acids, all that I was telling you. Essential amino acids are those which have to be supplied through diet, and non-essential amino acids are those which are synthesized in the body itself, right? Okay. Now, what we have to understand here, remember, human body can synthesize only 10 amino acids. Okay, these 10 amino acids which the body can synthesize, they are called non-essential amino acids. Okay, and they are also called as dispensable amino acids. While the remaining 10, I told you 20, is it not? The remaining 10 which the human body cannot synthesize, they are called non-essential amino acids or indispensable amino acids. Okay, so these essential amino acids are required for the growth of the body, and their deficiency, it causes many diseases. You must have heard about marasmus and kwashiorkor. Kwashiorkor is a protein deficiency disease. Okay, right. Now let us see structurally what these amino acids are. And I gave you the classification also: neutral amino acids, acidic amino acids, and basic amino acids, and also sulfur containing amino acids. Now this is the basic structure of amino acid. You know it, is it not? NH2, CH, R is attached, COOH. Now if this R is hydrogen, then you call it as glycine. Okay, if this R group is hydrogen, then it is called glycine. Okay, so here all the amino acids are given. I'll just explain a few. See, this is the general way of expressing any amino acid. Okay, now in this, the R is going to be replaced. If it is replaced by hydrogen, it is called glycine. If this R is replaced by a CH3 group, it is called alanine. And if it is replaced by an isopropyl group, then it is called valine. Now this is, what is this called? This is called as 2-methyl propyl, is it not? 2-methyl propyl attached to the CH group. Then it is called leucine, right? Then leucine and isoleucine. This is 2-methyl propyl, whereas this is 1-methyl propyl. Okay, this you have to number the carbon chain from here, from where the branch is starting. Okay, so this will be 1, 2, 3. So it is 1-methyl propyl, and this is 2-methyl propyl. Okay, 2-methyl propyl is leucine and 1-methyl propyl is isoleucine. Same way we saw alanine. Alanine is what? 2-methyl. Okay, this is 2-amino propanoic acid, you can call it, right? Now in this, the CH3 group, if it is replaced by one hydrogen replaced by phenyl group, then it is called as phenylalanine. Okay, so I'm just giving you a few just to explain what is the basic structure. Okay, and no need to worry that I have to learn all the 20 structures. You need not have to worry about it. But if you know a few, at least say around 4 or 5 with structures, if you know it, it will be good for you, right? So that is about the structure of amino acids. So I'll give you the picture of this. You can see it. Okay. Now let us come back. These essential amino acids, which are 10 in number, where do you find them? You find them in meat, fish, milk, eggs. So they are all called as complete proteins. These items, food items, they are considered to be complete proteins because they give all types of amino acids. Plant proteins are generally incomplete proteins like rice, wheat, corn, etc. When you say they're deficient in one or more amino acids, they will not give you all the amino acids which are required for your body. That is why you have to, if you take rice, then you have to take something else also to complement the other amino acid which may be present in that, is it not? You can't be taking only rice. That will not be sufficient for your body. Okay. Now I have told you about the properties of amino acid. It is showing amphoteric behavior. It is soluble in water, and it is existing as a dipolar ion, and that is called as a Zwitter ion. All that I have already discussed with you. I have told you, right? Now let us see what is meant by isoelectric point or isoelectric pH. It is that pH at which there is no net migration of amino acid under the influence of an applied electric field, right? It is that pH of the medium at which this amino acid is neither moving towards the cathode nor moving towards the anode. It does not move. There is no migration under the influence of an electric field. Okay. Now, what happens? Supposing the medium is acidic, then what will happen? This amino acid exists as a cation. Why? You see, when H plus is added, this H plus will go and get itself attached to NH2 group, which is basic in nature, and it will become NH3 positive. Now this whole unit is acting as a cation because of positive charge. So if it is a cation, it will move towards a negative electrode in the electric field, applied electric field, that is cathode. And supposing if the medium is basic, then what will happen? The COOH will give a proton and it will go with this OH minus as water. And what will be left behind? It will be left behind with COO minus, which is an anion. An anion is negatively charged, so it will go towards a positive electrode, that is anode. Okay, so at isoelectric pH, amino acid exists as a neutral dipolar ion. That's why there is no migration. Okay, there is no migration at the point of isoelectric pH, right? Okay, now